Hi, my name is Niveda Balendra from Royal West Academy, Montreal, Quebec. So after the H1N1 virus, there was a great demand for hand sanitizers. The main ingredient in hand sanitizers is ethanol. Not only hand sanitizers, but any antibacterial product, the main ingredient for the majority of these products is ethanol. So I really wanted to know whether it was the best there is out there, or whether there was an antibacterial agent that killed bacteria more efficiently than the products that we are using currently. So that was my main question, was to see if there was an antibacterial agent that was more effective than ethanol. So before starting to explain the experiment, just a few concepts I would like to touch upon. Ethanol, as I said, was the main ingredient in the majority of antibacterial products, but they're used at a great concentration of approximately 60 to 70%. Isopropanol is the second type of alcohol which I use to compare to ethanol. This is used in the majority of cleaning products, but also equally in alcohol swabs at a high concentration of 60 to 70%. If we see, both types of alcohol have similar structures. They differ by a few hydrogen and carbon atoms. They also have very similar properties. The type of bacteria which I tested on is E. coli, specifically the DH5 alpha strain. And the skin cells that I tested on are Hackett. These cells are keratinocytes, which we can see right here. And keratinocytes represent 95% of the epidermis, which is the outermost layer of our skin. So the objective of my experiment was to determine whether ethanol or isopropanol was more effective at killing bacteria and was less detrimental to the skin cells. So I split up the project into two parts. Part one consisted of testing which alcohol was more effective at killing bacteria. After concluding the results, this led me into the skin cells part of the project because essentially whenever we use these products, it is our epidermis that comes into contact with the product. So I really wanted to know whether it was harmful to our health or not. So part one, my hypothesis was that if the E. coli cells, DH5 alpha, are exposed to higher concentrations of ethanol, 50% and above, because the majority of hand sanitizers or alcohol swabs are at a great concentration, then I hypothesized that the bacteria will die. The second hypothesis was that if the cells, the bacteria, are exposed to various concentrations of isopropanol, then it would kill more bacteria than ethanol, so it will be more efficient. This was actually backed up by research done on surfaces, for example, where they exposed the surface and the bacteria on the surface to ethanol and isopropanol turned out to be the better, more effective agent. So that was my educated guess behind this. So the procedure in doing so, I prepare liquid cultures of E. coli with the alcohol. So as we can see here, these are my representations of the test tubes. So here is the ethanol, isopropanol, and I created 5 concentrations of each alcohol. Now, I prepared liquid cultures just to avoid the alcohol from actually evaporating, because if the alcohol evaporated, then the results would be flawed. Second, I incubated the prepared liquid cultures in agar plates. Now, this was to serve as a visual representation of the bacteria and to quantify the cell growth and to actually see the cells. And 2 trials were conducted for each concentration to ensure the precision of the results. Next, I took an absorbency test to quantify the cell growth. So here we can see a cuvette. Basically what I did is I added the liquid cultures into each cuvette, and initially a light is passed through. Depending on how much bacteria there are in the cuvette, whether there's more or less, it will absorb or retain the light that is initially passed through the cuvette, and whatever is left is transmitted to the end. So therefore, the higher the absorbance, the more bacteria. So here are my results. Now this test was to kind of optimize the concentrations that I was going to use in the next few experiments. So here we can see at 0, the plus represents growth of bacteria, and the negative represents no growth. So 0 and 10%, we can see that the bacteria grew, but 25 on, the bacteria didn't grow. Now I know for sure that I want to use 0 and 10% because I need bacteria to work with in order to compare ethanol with isopropanol. Since I use 0 to 10%, and I don't know anything, I don't know what's going on between 10 and 25, in order to maintain that equal concentration, I picked 10 to 20 as well. So my range now is 0 to 20%. Here's the absorbency test. Blue represents before incubation, and red represents after incubation. Now with the absorbency test, especially when there's E. coli cells with alcohol, it can easily become cloudy, and this would kind of change the results just by a few values. But nevertheless, the results are still precise because I conducted two trials for each. So if we compare both, we can see that the bacteria died before and after incubation. And from the agar plates, I said that I wanted to use 0 to 10%, 0 to 20, sorry. And we can see that the bacteria didn't really differ in those concentrations. So here are the values that I got from the agar plates. This is ethanol, and this is isopropanol results. So 1x represents the actual concentrations which I used. 10x is I diluted it 10 times, and 100, I diluted it furthermore 100 times. Now this was so I can have as many points as possible to work with and to possibly compare, because I can't possibly compare it with only the 1x. So here we can see that the bacteria, the inhibition rate, basically the inhibition rate is the capacity, in this case, of the alcohol to inhibit or stop the bacteria from growing. So we can see that it's increasing exponentially, and here too it's increasing. So the capacity of it to actually kill the bacteria more and more is increasing significantly. Here we can see that we can notice there's like a random big percentage here. Now the fact for this is because when I was actually preparing the plates, I must have not shaken the test tube. So if I didn't shake it, it formed a precipitate. And forming a precipitate, when I actually extract the bacteria, I'm not actually taking the bacteria, which is why 88.5% of the plate was not filled. So here's a graph that clearly demonstrates the inhibition rate of ethanol versus isopropanol. Ethanol is the blue line, and isopropanol is the red line. So we can clearly see that isopropanol reaches 20% before ethanol. Even though this is a small difference, it is still significant in that we always thought that ethanol was the more effective antibacterial agent. Here we can also see that the red line, which is isopropanol still, reaches 100% at about 17.5% concentration, and ethanol reaches it at about 19.8. Again, this is a small percentage difference, but it is still significant because the fact is that isopropanol is consistently more effective than ethanol, even at lower concentration. This also adds to the fact that we shouldn't even be using high concentrations of alcohol in any antibacterial product. In fact, we can use lower concentrations. Part 2 of my experiment were the skin cells. So my hypothesis was that if the skin cells are exposed to various concentrations of ethanol, knowing now that it was less harsh than isopropanol towards bacteria, my hypothesis was that the skin cells would survive, the majority of them. So the second hypothesis was that if the skin cells are exposed to concentrations of isopropanol, then the majority of skin cells would die, as opposed to ethanol that would have more skin cells left compared to isopropanol. In order to do so, I prepare liquid cultures, again, with the serum, which is the nutrient for the skin cells, plus the alcohol. So I prepared 5 concentrations, the same exact concentrations, just to ensure the consistency throughout the whole experiment. After this, I took the cells out of the incubator, which were already incubated at 37 degrees Celsius for 48 hours. And we must note that skin cells take more time to grow than bacteria. So we took it from the 6-well plate, and then basically it already had solutions in it because the nutrients from beforehand. And then I extracted all the solutions. I cleaned it with PBS solution. And then I added the prepared liquid cultures with the concentrations. And here is an example with already the concentrations inside. And after that, I incubated at 37 degrees Celsius for 48 hours. After this, after incubating it, I took it out and I extracted all the solution. And then I added the trypsin. Now what trypsin, which is right here, it does, it basically cleaves the protein and resuspends the cells. After this, I added it to the grid slide, and then I put it under the microscope. And basically what we see is that we compare both of these quadrants, and then when we compare both of these quadrants, the cells that are in there, and we take the average of the cells, and this represents how much cells are in the plate. And here is just a visual representation of these cells. So my results. Now here are the results, and we can see that, for example, at 20%, the inhibition is 99.8, and here it's 86.5. So we see that isopropanol killed less skin cells than ethanol. Here is a clearer graph. If we follow the trend line, for example, from the first experiment, I suggested that we use about 17.5% if we rounded 20%. Here, 20%, it kills 50% of the skin cells, whereas ethanol kills all the skin cells beforehand. So my first hypothesis from the first experiment was right in that isopropanol proved more effective than ethanol. Second experiment, I was actually surprised to see that the more harsh antibacterial agent wasn't as harsh on skin cells. It actually killed less skin cells. So in conclusion, isopropanol is a better antibacterial agent. And by reducing the concentration in these products, it will also reduce the toxicity in the bloodstream. Because according to research, whenever we use such products, it's absorbed through the skin, and it increases the toxicity levels in our bloodstream. So by reducing the concentration, we're also reducing the risk of toxicity in our bloodstream. It is also a cheaper alternative. Now this adds to, not only is there a health aspect, but there's always the economical aspect. And knowing that it is a cheaper alternative is very important. It is also less harmful to the epidermis. All in all, a lot of people consume these products. In a hospital setting, doctors, nurses, patients, they use it on a day-to-day basis and multiple times per day. Especially injection sites, diabetic patients, children use it. Parents really want them to use the products. So it really shows that it will really change the products and the way that we use them if we just simply change the ingredient that we use. So I think it is time to say goodbye to ethanol and hello to isopropanol. Thank you.
Thank you very much, Namata.
And bibliography. Yeah.
Thank you very much, and thanks for sticking to the time.
Thank you.
That's all. Any questions of Namata?
Well, my first question is very general. You went very quickly through the acknowledgement slide, and I guess the question I have is where did you conduct the work?
Oh, of course. Sorry. I didn't know if I had enough time.
Okay.
I conducted the research at the McGill University Health Center. As you noticed, there are 2 parts to the project. So part 1 was under the guidance of Carlos Melendez Pena. He, at that time, was working at the Immunology Center at Montreal General Hospital. And then for the skin cells part of it, I was working under Dr. Carter Lee, again at the Plastic Surgery Research Center at Montreal General Hospital.
Thank you.
Thank you.
So given that you use they already use isopropanol in wipes, but they don't use it in hand sanitizer, well, how do you think that happened? Because they obviously had the data to say isopropanol works in the wipes, although I agree at the higher concentration. So why do you think it happened that we use it for one thing and not the other thing?
Well, that's a very good question, especially since it's equally used. Ethanol is equally used in alcohol swabs and hand sanitizers, but isopropanol is used in comparison to both in alcohol swabs. I mean, I really wouldn't know the exact answer, but the miscibility properties are like isopropanol is just as miscible in water as ethanol is, and it is also less dense than ethanol is. So it has practically the same properties.
Yes, I would like to know exactly what your research brings that was absolutely not known, because obviously if you chose isopropanol, it's because there already there were indications that it was a good antibacterial. So, what's new in your research that wasn't known before?
Okay. Well, the fact is that the previous research, which my mentor sent me after I conducted the experiment, he told me that, well, there are a few people who did around the same experiment, but the procedure varied. Also, they tested on surfaces, but like my main goal behind it was for health purposes and to see how it also, that's why I added the skin cells part of it, which I didn't see anyone who actually tested on skin cells. And I really think it's applicable to society right now because these are products that are widely used right now by patients, by a large society of, a large group of consumers. Sorry.
Just a short follow-up. When isopropanol is used in the various cleaners and stuff, what concentration is it?
It's also at high concentration.
It's also high concentration.
Yeah, a couple of questions. Can you tell me why you chose that particular strain of bacteria, DH5 alpha? Is there a particular reason why that was chosen?
Well, E. coli cells are the most common bacteria to use in laboratory experiments. Also, it was widely available, especially for my mentor. And I do want to furthermore try it on different strains of bacteria. But, I mean, they are harmful bacteria like MRSA or Staphylococcus or any other type of bacteria. But really, it's also one of the harder types of bacteria to kill out of the gram-negative. And it's also, as I said before, widely used in experiments and just a kind of a prototype bacteria to use.
Okay. And one other thing, your experiments with the bacteria, you grew them for 24 hours. And bacteria grow very rapidly. So after 24 hours, you've probably got what's called a saturated culture. Did you do, or would you contemplate doing a time course and maybe compare the growth rates under the various conditions? And have you done that, or and just not reported it? And what kind of what might you expect? And if you've done it, what did you get?
Well, that's a very interesting add to the project that I would for sure want to do in the future. For now, I concentrated on just the basic 24-hour time. But definitely, I mean, whenever we use products, there's also the difference of time that we actually expose it to our skin, for example, or the bacteria. So it would definitely be a really good add to the project if we can actually see the different time frames. But for this time, no, I used only 24 hours. And sorry, just to add to the E. coli, I just remembered, it's also present on our skin. So it's a really good type of bacteria to use for this reason.
Thank you, just one or two. Did you have a question? Hi, I just have a short question, I guess, about your last graph. Maybe you could pull that up.
Okay.
About the skin cells. I noticed that your data point for, most of your data points for the,
Oops, sorry.
it's okay, it can be small, I guess. I noticed that most of your data points for the isopropanol are pretty similar to the ethanol, and then there's just one outlier at the 50% that really skews the line. And if we took out that outlier, it would look to me that at these lower concentrations of 0 to 20% that you've been talking about, that isopropanol and ethanol essentially have the same effect on inhibition of human skin cells. So I'm wondering, is that just an outlier? How many replicates did you do of this experiment? Like, why did you choose a linear fit for this data? And also, where is the 50% data point for ethanol?
Okay, so basically I chose the linear graph because I thought that it was following, especially if we see ethanol, it's following a linear line. So I thought, I know for sure that I wanted to use a trend line because whenever we're comparing to alcohols in this case, it's not right to follow the points because there's always the approximate that we had to take. So that's why I plotted a trend line. I picked the linear line because I believe that it was following a linear trend. But definitely about the outlier. I mean, even if we did, as you said, it would have been the same. But if we actually look at the points beforehand, there's also always like kind of a minimum 10% difference between, if these are the actual points, if there's always a minimum of 10% difference between the inhibition rate of ethanol and isopropanol. Now 10% is still considerably more. It's not like 1% or 2% of skin cells left alone. That's 10% of whatever area we're covering, which is possibly millions of cells, which are still left unaffected by the alcohol.
And so just, I guess, how many replicates of the experiment did you do?
I did 3 replicates of the
And the 10%
experiment.
is statistically significant. Okay, thanks.
Thank you.
Okay, second last question, and I have one.
Okay. Maybe a little bit further along those lines, I think application of statistics to the lines would help, and that would tell us if they actually were significantly different. Do you see other applications for this beyond the clinical setting?
I mean, apart from, sorry, what do you mean?
Beyond hospitals and patient settings, those types of things. Where would you see this having utility?
I mean, like, a lot of people use it outside of the hospital, especially after the H1N1 virus. There was a high demand for hand sanitizers, for example. A lot of children use it, and especially since they're developing their immune system, and to actually impose a higher concentration of ethanol. And you really want to be sure that you're getting rid of all the bacteria, especially with the toxicity levels that I was talking about. A lower concentration is really better, especially for children. And it's not only in a hospital setting, but essentially anywhere that we use antibacterial products at home. For example, my grandmother, she was a diabetic patient. She always injected multiple times per day, insulin, per day, 2 times per day. So she used alcohol swabs always. My mom too. So it's really prevalent not only at the hospital but also at home.
Okay.
Just a quick comment. In your conclusion, you talked about the killer effect of this on the cells. You talked about the cheap alternative and less harmful to the epidermis. You also mentioned that it was less toxic to the bloodstream. I presume that is from the literature, am I correct?
Mm-hmm.
What would you do to show if in fact it is less toxic to the bloodstream if you in fact had another couple of months to do this exercise?
I mean, I would surely read over the previous research that has been done, and if possible, if they had a procedure to do so. But I mean, I haven't really gone into the bloodstream portion of it, but I wouldn't know if we were allowed to test on human beings or not. But knowing that it is only a minimal toxicity level, I mean, we could possibly tell them to use it over a certain period of time and then we can actually test the toxicity levels of the bloodstream, comparing ethanol to isopropanol.
And now let's assume that these are really great findings, which I'm sure they are.
Thank you.
How would you go about commercializing your findings?
I would surely make sure it's cheaper than ethanol, because, I mean, if we see Purell, for example, the hand sanitizer are really expensive and not everyone can afford it, just for one bottle, especially considering how much we actually use it per day on week-to-week basis. The bottle will finish really soon. I mean, since isopropanol is a cheaper alternative, the price would also decrease, and the consumers would also be more happy to realize that their skin isn't being affected as much. And that's essentially it, just to make sure that the consumers in society have an alternative, aren't being fooled that it actually kills all of the cells, but also the majority of their skin cells. But that they really get what they pay for, which is really important at the end of it. More than the economical aspect, I prioritize the health aspect.
Was this your idea? Whose idea was this? This was yours?
This is my idea.
Not the mentor's idea. Very nice. Thank you very much.
Thank you
for
very
being
much.
here.
Thank you.
Appreciate your time.
Thank you very
Which grade
much.
are you in, by the way?
I am in grade 11.
Okay. Thank you very much for being
Thank you
here.
very much. Thank you. Thank you.